This section introduces glycogen, a critical molecule for energy storage in the body. Glycogen is a highly branched polysaccharide composed of glucose units. It serves as the primary storage form of carbohydrates in animals. Glycogen has a unique branching structure. It consists of glucose units linked together in chains with multiple branching points. Glycogen serves three primary functions in the body. First, glycogen acts as an efficient energy storage system. The body converts excess glucose into glycogen for storage, similar to charging a battery for later use. Second, glycogen plays a crucial role in regulating blood glucose levels. When blood sugar drops between meals, the liver breaks down glycogen to release glucose, maintaining steady levels in the bloodstream. Third, glycogen stored in muscles provides immediate energy during physical activity. This allows for rapid energy release without relying on the bloodstream, making it essential for exercise performance. To summarize, glycogen is a highly branched polysaccharide that serves as the body's primary carbohydrate storage form. It plays essential roles in energy storage, blood glucose regulation, and providing fuel for physical activity. Glycogen has a complex branched structure that allows for efficient glucose storage and release. The basic building block of glycogen is glucose, which forms a compact hexagonal structure. In glycogen, glucose units connect through alpha-1,4 glycosidic bonds to form linear chains. What makes glycogen unique is its branching structure. Branch points connect through alpha-1,6 glycosidic bonds, creating a tree-like molecule. This branch structure creates a compact molecule that resembles a tree, with many endpoints available for rapid glucose release. This branching structure provides several advantages. Multiple endpoints allow enzymes to release glucose simultaneously from different branches. The compact design increases solubility and maximizes storage efficiency. In summary, glycogen's branched molecular structure provides the perfect balance between compact storage and rapid glucose mobilization, making it an ideal energy reserve for the body. Glycogen, the primary carbohydrate storage molecule in the human body, is stored in two main locations. The first major storage site is the liver, which contains approximately 100 grams of glycogen. The second and larger storage site is skeletal muscle, which holds approximately 400 grams of glycogen. These storage locations contain different amounts of glycogen, with muscles storing 4 times more than the liver. Despite storing less glycogen, the liver plays a crucial role in maintaining blood glucose levels. When blood glucose falls, liver glycogen is broken down and released into the bloodstream. In contrast, muscle glycogen serves a different purpose. It provides energy directly to muscle cells during physical activity, especially during high-intensity exercise. Unlike liver glycogen, muscle glycogen cannot be released into the bloodstream. It can only be used by the muscle cells themselves, which makes it a local energy reserve rather than a contributor to blood glucose regulation. This key difference in function reflects how the body has evolved specialized storage systems to meet different metabolic needs. Glycogen stored in the liver plays a crucial role in maintaining stable blood glucose levels throughout the day. The liver contains approximately 100 to 120 grams of glycogen in healthy adults, serving as our primary glucose reservoir. Unlike muscle glycogen, which can only be used by the muscles themselves, liver glycogen can be broken down and released as glucose into the bloodstream. This graph shows how liver glycogen helps maintain blood glucose levels within the normal range throughout the day, even between meals and during overnight fasting. When blood glucose levels begin to fall, the liver initiates glycogenolysis, the breakdown of glycogen into individual glucose molecules. Enzymes like glycogen phosphorylase cleave glucose units from the glycogen branches, starting from the outer ends. Unlike skeletal muscles, liver cells contain the enzyme glucose-6-phosphatase, which allows them to release free glucose directly into the bloodstream. This release of glucose from liver glycogen is especially important during overnight fasting when we go many hours without consuming food. Without liver glycogen, our blood glucose levels would drop dangerously low during sleep, potentially leading to hypoglycemia. Let's review the key points about liver glycogen's role in glucose homeostasis. The liver stores about 100 to 120 grams of glycogen in adults, which can maintain blood glucose for approximately 24 hours of fasting. Unlike skeletal muscles, liver cells possess glucose 6-phosphatase, allowing them to release free glucose directly into the bloodstream. Liver glycogen metabolism is tightly regulated by hormones like insulin and glucagon, ensuring blood glucose remains within the normal range. This system is especially critical overnight when we go 8 to 12 hours without food, preventing dangerous drops in blood glucose. In skeletal muscles, glycogen serves as a critical energy source during physical activity. Skeletal muscle fibers store glycogen as granules distributed throughout the sarcoplasm. Glycogen in muscles has a highly branched structure, allowing for rapid mobilization during exercise. During exercise, muscle glycogen is broken down in a process called glycogenolysis to provide energy. This process ultimately leads to ATP production, which fuels muscle contraction. The contribution of glycogen to energy production increases with exercise intensity. During high-intensity exercise, muscle glycogen becomes the predominant fuel source, providing up to 90% of the energy. Unlike liver glycogen, muscle glycogen can only be used within the muscle itself. This is because muscle cells lack the enzyme glucose 6-phosphatase, which is needed to release free glucose into the bloodstream. Understanding how muscle glycogen is utilized is crucial for optimizing exercise performance and recovery. Glycogenesis is the metabolic pathway for converting glucose into glycogen for storage in the liver and muscles. Step 1 of glycogenesis is glucose phosphorylation. The enzyme hexokinase or glucokinase in the liver transfers a phosphate group from ATP to glucose, creating glucose 6-phosphate. In step 2, the enzyme phosphoglucomutase converts glucose 6-phosphate to glucose 1-phosphate by moving the phosphate group from the 6th carbon to the 1st carbon position. In step 3, UDP glucose pyrophosphorylase converts glucose 1-phosphate to UDP glucose using UTP as a co-substrate. Then in step 4, the enzyme glycogen acts as a primer, initiating glycogen synthesis by adding the first glucose molecules to itself, creating a short initial chain. In step 5, glycogen synthase extends the glycogen chain by adding glucose units from UDP glucose to the non-reducing end of the growing chain, forming alpha-1,4 glycosidic bonds. In the final step, the branching enzyme transfers a segment of about 7 glucose units from the growing chain to create a branch point. This creates alpha-1,6 glycosidic bonds at branch points, giving glycogen its characteristic branched structure. This branched structure allows glycogen to store a large amount of glucose in a compact form while providing multiple sites for rapid glucose release when needed. This section explores the 5 critical enzymes involved in glycogen synthesis. The glycogen synthesis pathway involves a series of enzymatic reactions. The first enzyme, hexokinase or glucokinase, phosphorylates glucose to form glucose 6-phosphate. This step traps glucose inside the cell by preventing it from crossing the cell membrane. Next, phosphoglucomutase converts glucose 6-phosphate to glucose 1-phosphate by relocating the phosphate group from the 6th carbon to the 1st carbon position. The third enzyme, UDP glucose pyrophosphorylase, activates glucose 1-phosphate by attaching UDP, forming UDP glucose, which serves as the active donor for glycogen synthesis. Glycogen synthase then transfers glucose from UDP glucose to the growing glycogen chain, forming alpha-1,4 glycosidic bonds. This is the rate-limiting enzyme in glycogen synthesis. Finally, the branching enzyme transfers segments from the linear chain to create branch points by forming alpha-1,6 glycosidic bonds. These branches increase glycogen's solubility and create multiple sites for rapid synthesis and breakdown. Together, these 5 enzymes work sequentially to build the complex, branched glycogen structure that serves as the body's primary carbohydrate storage form. Understanding these enzymes is crucial for comprehending glycogen metabolism and its disorders. Glycogenolysis is the metabolic process by which glycogen is broken down to provide glucose during times of need. To understand this process, let's first look at the structure of glycogen, which consists of glucose units connected by alpha-1,4 linkages in the main chains and alpha-1,6 linkages at branch points. Step 1 of glycogenolysis involves glycogen phosphorylase, which cleaves glucose units from the non-reducing ends of glycogen chains. When phosphorylase reaches a branch point, it stops. This is where the debranching enzyme comes in, with its dual functions. In the final step, the glucose 1-phosphate produced by phosphorylase is converted to glucose 6-phosphate, which can enter glycolysis or be released into the bloodstream. To summarize, glycogenolysis is a well-regulated process that efficiently breaks down glycogen's complex structure to provide glucose when the body needs energy. 4 key enzymes work together to break down glycogen into glucose molecules that can be used by cells or released into the bloodstream. Glycogen phosphorylase is the first and rate-limiting enzyme in glycogenolysis. It cleaves the F1,4 glycosidic bonds between glucose units, releasing glucose 1-phosphate. The debranching enzyme handles the branch points in glycogen. It has 2 activities: transferase activity that relocates glucose units, and alpha-1,6 glucosidase activity that cleaves the branch point, releasing free glucose. Phosphoglucomutase converts glucose 1-phosphate produced by glycogen phosphorylase into glucose 6-phosphate. This is a necessary step because glucose 6-phosphate can enter glycolysis for energy production. In the liver, but not in muscle, glucose 6-phosphatase removes the phosphate group from glucose 6-phosphate, producing free glucose that can be released into the bloodstream to maintain blood glucose levels. In muscle cells which lack glucose 6-phosphatase, glucose 6-phosphate is directed into glycolysis for ATP production to power muscle contractions. Together, these 4 enzymes coordinate to ensure efficient glycogen breakdown, providing glucose for cellular energy needs or for maintaining blood glucose levels. Glucagon is a hormone with effects opposite to insulin, primarily promoting glycogen breakdown. Glucagon is secreted by the alpha cells of the pancreas during fasting states when blood glucose levels fall too low. When blood glucose levels fall too low, such as during fasting or between meals, alpha cells in the pancreas detect this drop and respond by secreting glucagon. Glucagon enters the bloodstream and travels to the liver, its primary target organ. Once glucagon reaches the liver, it triggers a signaling cascade that affects glycogen metabolism. Glucagon has 2 major effects on enzymes involved in glycogen metabolism. The net result is rapid glycogen breakdown in the liver and release of glucose into the bloodstream. This process raises blood glucose levels back to normal, counteracting hypoglycemia and providing energy during fasting states. Intracellular signaling in glycogen regulation involves complex cascades that translate hormonal signals into enzymatic actions. When hormones like glucagon, epinephrine, or insulin bind to their receptors, they initiate signaling pathways that ultimately affect glycogen metabolism. The first pathway we'll examine is the CAMP pathway, triggered by hormones like glucagon and epinephrine. When glucagon or epinephrine binds to a G-protein-coupled receptor on the cell surface, it causes a conformational change in the receptor. This activates a G-protein, which in turn activates the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP, which acts as a second messenger. CAMP activates protein kinase A, or pKa, by binding to its regulatory subunits and causing the release of catalytic subunits. Activated pKa phosphorylates and activates glycogen phosphorylase, which breaks down glycogen, while also phosphorylating and inhibiting glycogen synthase. A second important pathway is the calcium signaling pathway, which is activated by epinephrine binding to alpha receptors. When epinephrine binds to an alpha receptor, it activates phospholipase C, or PLC, through a different G protein. PLC cleaves phosphatidylinositol 4,5-bisphosphate to produce inositol triphosphate, or IP3, which diffuses into the cytosol. IP3 triggers the release of calcium ions from the endoplasmic reticulum, increasing cytosolic calcium concentration. Calcium binds to calmodulin, forming a calcium-calmodulin complex. This complex activates phosphorylase kinase, which then phosphorylates and activates glycogen phosphorylase, leading to glycogen breakdown. The insulin signaling pathway contrasts with the previous pathways by promoting glycogen synthesis rather than breakdown. Insulin binds to the insulin receptor, a receptor tyrosine kinase, causing receptor autophosphorylation. The activated receptor phosphorylates insulin receptor substrates, or IRS proteins. IRS proteins activate phosphatidylinositol 3 kinase, or PI3K. This leads to the activation of protein kinase B, also known as ACT. ACT activates protein phosphatase 1, or PP1, which dephosphorylates glycogen synthase, activating it, while also dephosphorylating glycogen phosphorylase, inactivating it. In summary, intracellular signaling pathways coordinate the regulation of glycogen metabolism. The C-AMP and calcium pathways triggered by glucagon and epinephrine promote glycogen breakdown. In contrast, the insulin pathway promotes glycogen synthesis, storing excess glucose for future energy needs. These opposing pathways ensure precise control of glycogen metabolism in response to changing energy demands and nutritional states. The glycogen shunt is a metabolic pathway where glucose passes through glycogen before entering glycolysis. Even when glucose is abundant, some glucose molecules cycle through glycogen formation and breakdown before continuing to glycolysis. Let's visualize the glycogen shunt pathway. The glycogen shunt involves multiple steps. First, glucose is phosphorylated to glucose 6-phosphate. Then, glucose 6-phosphate is converted to glucose 1-phosphate. Glucose 1-phosphate is activated to UDP glucose, which is incorporated into glycogen. Shortly after, glycogen phosphorylase breaks down glycogen back to glucose 1-phosphate. Finally, glucose 1-phosphate returns to glucose 6-phosphate and enters glycolysis. Despite appearing inefficient, the glycogen shunt offers several metabolic advantages. First, it enhances control of glycolytic flux by adding another regulatory step. The shunt helps regulate glucose 6-phosphate concentrations in the cell. It maintains glycogen turnover, keeping glycogen metabolism active. Studies show the shunt can improve insulin sensitivity in muscle cells. Finally, it prevents excessive activation of glycolytic and related pathways. Let's examine how the glycogen shunt helps regulate glucose metabolism. Glucose 6-phosphate sits at a metabolic crossroads where it can enter glycolysis, the pentose phosphate pathway, or the glycogen shunt. The glycogen shunt demonstrates that what appears metabolically inefficient can actually provide important regulatory advantages for cellular energy metabolism. Glycogen supercompensation is a physiological process where muscle glycogen stores are increased beyond normal levels through specific exercise and dietary protocols. Let's compare normal glycogen levels to what happens during supercompensation. The normal glycogen level in muscles serves as our baseline at 100%. During the supercompensation protocol, glycogen levels first decrease significantly during the depletion phase, often dropping to 40 to 60% of normal levels. Then, during the loading phase, with increased carbohydrate intake, glycogen levels not only recover but exceed normal levels, reaching up to 160% of baseline. This peak represents the supercompensation effect. The supercompensation protocol typically follows 3 main phases, spread over about a week. Days 1 to 3 focus on glycogen depletion through high-intensity exercise and a low-carbohydrate diet containing just 50 to 100 grams per day. This depletes muscle glycogen stores. During days 4 and 5, the loading phase begins with reduced training intensity and a high-carbohydrate diet of 7 to 10 grams per kilogram of body weight. This rapidly restores glycogen levels. Finally, in days 6 and 7, the supercompensation phase involves minimal exercise, continued high carbohydrate intake, and allows glycogen stores to exceed normal levels. Let's examine the physiological mechanisms that make glycogen supercompensation possible. Following glycogen depletion, several adaptive mechanisms occur. First, glycogen synthase activity increases significantly. Second, muscle cells experience enhanced insulin sensitivity, allowing for greater glucose uptake. Third, there's an upregulation of GLUT4 glucose transporters on the cell membrane, further facilitating glucose entry. Finally, the muscle develops an expanded storage capacity, allowing more glycogen to be stored than under normal conditions. Glycogen supercompensation is widely applied in endurance sports where performance depends on sustained energy availability. The benefits include a 2 to 3% performance improvement, which is significant at elite levels. Athletes also experience extended time to exhaustion, delayed fatigue, and can maintain a higher work rate throughout competition. However, there are limitations to consider. The effect is temporary, lasting only 5 to 7 days. Athletes may experience digestive discomfort during the loading phase, and the technique offers little benefit for short-duration, high-intensity activities. GSD type 1, also known as von Gierke disease, is a glycogen storage disorder caused by a deficiency in glucose 6-phosphatase. The pathophysiology involves a missing glucose 6-phosphatase enzyme, which prevents the conversion of glucose 6-phosphate to glucose in the liver. Without this enzyme, glucose cannot be released from the liver into the bloodstream, leading to glycogen accumulation in the liver and kidneys. This disorder presents with several distinct clinical manifestations. Diagnosis of von Gierke disease involves several different approaches. Management strategies for von Gierke disease focus on preventing hypoglycemia and managing complications. With proper management, patients with von Gierke disease can lead relatively normal lives, although careful monitoring is required throughout their lifetime. Pompe disease is a rare genetic disorder caused by mutations in the GAA gene. This results in deficiency of the acid alpha-glucosidase enzyme, which is essential for breaking down glycogen in lysosomes. The disease follows an autosomal recessive inheritance pattern. In normal cells, the enzyme acid alpha-glucosidase breaks down glycogen in lysosomes. In Pompe disease, this enzyme is deficient or absent, resulting in glycogen buildup inside lysosomes. Pompe disease presents with two main forms, based on age of onset and severity. The infantile onset form is most severe, with symptoms appearing shortly after birth. It causes severe heart enlargement, muscle weakness, and is fatal within the first few years without treatment. The late onset form may appear in childhood or adulthood and progresses more slowly. It primarily affects skeletal muscles, especially in the legs and respiratory system, but usually spares the heart. Enzyme replacement therapy is the primary treatment for Pompe disease, involving regular infusions of recombinant acid alpha-glucosidase. This therapy has significantly changed the outlook for patients. Benefits include prolonged survival, improved cardiac and skeletal muscle function, and enhanced motor development. However, ERT has limitations. It's extremely expensive, may trigger immune responses, doesn't penetrate all tissues equally, and requires lifelong biweekly infusions. Enzyme replacement therapy for Pompe disease has evolved since the first approval of Myozyme in 2006. Research into Pompe disease continues with several promising approaches. These include improved enzyme replacements, gene therapy to restore GAA production, chaperone therapy, and combination approaches. Beyond the more common types previously discussed, several other important glycogen storage diseases significantly impact metabolism. Type 3 or Cori disease is caused by a deficiency in the debranching enzyme amylo-1,6-glucosidase. Patients with Cori disease present with hepatomegaly, growth retardation, hypoglycemia, and progressive muscle weakness. Management focuses on a high-protein diet, frequent meals to prevent hypoglycemia, and regular monitoring for liver dysfunction. Type 4, or Anderson disease, is caused by a deficiency in the branching enzyme, which prevents proper glycogen branching. Anderson disease presents with progressive liver cirrhosis, failure to thrive, hypotonia, and is often lethal in early childhood. Management is largely supportive, with liver transplantation considered in select cases. Type V, or McArdle disease, is caused by a deficiency in muscle glycogen phosphorylase, preventing glycogen breakdown in muscle tissue. McArdle disease is characterized by exercise intolerance, muscle cramps during exercise, the unique second wind phenomenon, and myoglobinuria after intense exercise. Management involves moderate exercise regimens, maintaining a high carbohydrate diet, and avoiding intense exercise that could trigger rhabdomyolysis. Type 6, or HERS disease, results from deficiency in liver glycogen phosphorylase, affecting the liver's ability to release glucose from glycogen. HERS disease presents with mild hepatomegaly, growth retardation, and mild hypoglycemia, but is generally milder than other glycogen storage diseases. Management includes frequent meals, cornstarch therapy to provide slow-release glucose, and monitoring growth and development. To summarize, these glycogen storage diseases differ in their enzyme deficiencies, affected organs, and clinical presentations, but all result from impaired glycogen metabolism. Glycogen serves as the primary fuel source during exercise, with its utilization pattern varying based on exercise intensity and duration. Exercise intensity dramatically affects how quickly glycogen is depleted from muscles. During low-intensity exercise, like walking or light jogging, muscles primarily use fat as fuel, with minimal glycogen utilization. As intensity increases to moderate levels, such as steady running or cycling, glycogen becomes a more significant energy source, accounting for roughly half of the energy requirement. High-intensity activities like sprinting or interval training rely heavily on glycogen, rapidly depleting stores due to the body's inability to mobilize fat quickly enough. A phenomenon known as hitting the wall occurs when glycogen stores become critically depleted during prolonged exercise. This typically happens around 90 to 120 minutes into high-intensity endurance exercise, when liver and muscle glycogen stores approach depletion. Athletes experience sudden fatigue, decreased performance, and mental confusion. Athletes employ several strategies to optimize glycogen availability for competition and training. Carbohydrate loading involves increasing carbohydrate intake for several days before competition, allowing muscles to store up to 50% more glycogen than normal. Strategic nutrition timing ensures carbohydrates are consumed before, during, and after exercise to maintain and replenish glycogen levels. Regular endurance training increases the muscle's capacity to store glycogen, improving overall endurance performance. Athletes also use glycogen-sparing techniques like fat adaptation and careful pacing to conserve glycogen during long events. The implications for athletic performance are significant. Endurance capacity is directly limited by glycogen availability, and higher exercise intensities deplete glycogen more rapidly. Proper glycogen management becomes especially critical for events like marathons, team sports with prolonged activity periods, and multi-day competitions where glycogen depletion can severely impact performance. Dietary carbohydrates significantly impact glycogen stores in the body. The quantity and quality of carbohydrates directly affect how much glycogen we can store. Higher carbohydrate intake typically leads to larger glycogen stores, while complex carbohydrates provide sustained glycogen synthesis. The fiber content of food affects absorption rate and glycogen formation. The timing of carbohydrate intake is crucial for glycogen synthesis. There are 3 key periods to consider: pre-exercise, during exercise, and post-exercise. Consuming carbohydrates 1 to 4 hours before exercise enhances glycogen availability. During exercise, simple carbohydrates help maintain blood glucose levels. After exercise, there's a glycogen window of 30 to 60 minutes when synthesis is optimal. For best recovery, athletes should consume 1 to 1.2 grams of carbohydrate per kilogram of body weight. Carbohydrate loading is a strategy used by athletes to maximize glycogen stores before competition. The graph shows how muscle glycogen levels change during a typical carb loading protocol. The classic protocol involves a 3-day depletion phase, followed by a 3-day loading phase. Modern protocols often skip the depletion phase and focus on 3 to 4 days of high carbohydrate intake, ranging from 8 to 12 grams per kilogram of body weight per day. This strategy can increase glycogen stores by 50 to 100% above normal levels, which is particularly beneficial for endurance events lasting more than 90 minutes. Different dietary approaches have varying effects on glycogen storage. Let's examine how carbohydrate percentage in the diet impacts glycogen levels. High carbohydrate diets containing 55 to 70% carbs maximize glycogen stores. Moderate carbohydrate diets provide adequate stores, while low carbohydrate diets significantly reduce glycogen capacity. Ketogenic diets with less than 10% carbohydrates result in minimal glycogen storage. When following a low carbohydrate diet, the body undergoes metabolic adaptation over 2 to 4 weeks. This adaptation reduces dependence on glycogen by shifting to fat utilization. While this may impair high-intensity performance, research suggests it can preserve endurance capacity after full adaptation. To summarize, carbohydrate quantity, quality, and timing all affect glycogen synthesis. The post-exercise glycogen window is crucial for recovery. Carbohydrate loading can dramatically increase glycogen stores for endurance events. Your dietary approach should match your performance goals and metabolic needs. And while low carbohydrate diets require adaptation, they can be viable for some athletes depending on their training and competition requirements. In normal physiology, insulin plays a crucial role in glycogen metabolism. When insulin binds to its receptors on liver and muscle cells, it triggers a signaling cascade. This promotes several key processes that regulate glycogen metabolism. In diabetes, glycogen metabolism is significantly altered depending on the type of diabetes. Type 1 diabetes is characterized by insulin deficiency due to autoimmune destruction of pancreatic beta cells. This leads to decreased glycogen synthesis and increased glycogen breakdown, contributing to hyperglycemia. In type 2 diabetes, cells become resistant to insulin signaling, despite normal or even elevated insulin levels. This insulin resistance impairs glycogen synthesis and leads to abnormal glycogen storage patterns in liver and muscle. Let's examine the molecular basis of insulin resistance and how it affects glycogen metabolism. In normal insulin signaling, insulin binds to its receptor and activates a cascade of proteins. This pathway ultimately activates glycogen synthase, promoting glycogen formation and storage. In insulin resistance, this signaling pathway becomes impaired at multiple points. Several mechanisms contribute to insulin resistance, reducing the cell's ability to synthesize and store glycogen properly. The relationship between diabetes, insulin resistance, and glycogen metabolism creates a vicious cycle. Insulin resistance leads to impaired glycogen synthesis in liver and muscle tissues. This impairment contributes to hyperglycemia, as glucose cannot be efficiently stored as glycogen. Chronic hyperglycemia causes glucotoxicity, which further damages cells and worsens insulin signaling. Finally, this glucotoxicity exacerbates insulin resistance, completing and reinforcing the cycle. This cycle has several pathophysiological consequences that worsen diabetic symptoms over time. Glycogen isn't just found in animals, it's also present in many microorganisms. Glycogen-like polysaccharides are found across bacteria, fungi, and even archaea. Microbial glycogen has a similar structure to animal glycogen, but with some key differences. Microbial glycogen typically has fewer branch points and lower molecular weight, though it maintains the same alpha-1,4 and alpha-1,6 linkage pattern. The presence of glycogen across different domains of life suggests it appeared very early in evolution. Glycogen represents an ancient solution to energy storage that has been conserved across billions of years of evolution, providing an adaptive advantage in fluctuating environments. Glycogen plays a crucial role in microbial survival during adverse conditions. Microorganisms store glycogen as a survival strategy for periods of nutrient limitation, dormancy, and during biofilm formation. Glycogen provides a rapidly accessible energy reserve, serving as both a carbon and ATP source that enhances microbial persistence in challenging environments. The essential role of glycogen in bacterial survival makes it an attractive target for new antimicrobial strategies. Targeting enzymes involved in glycogen metabolism, such as glycogen synthase GLGA, could disrupt bacterial survival mechanisms. Many pathogens rely on glycogen stores during infection processes. Disrupting glycogen metabolism could affect virulence and offers potential for developing new classes of antibiotics. Recent research continues to uncover new aspects of glycogen biology and potential therapeutic applications. As our understanding of glycogen metabolism evolves, several exciting research directions are emerging. Novel regulatory mechanisms are being discovered, including post translational modifications of enzymes, non coding RNAs, and the spatial organization of glycogen within cells. Research is revealing that glycogen serves roles beyond energy storage, functioning as a scaffold for proteins and influencing cellular stress responses. Advances in glycogen storage disease treatments include promising gene therapy approaches, improved enzyme replacement therapies, and novel small molecule chaperones. Emerging research explores connections between glycogen metabolism and neurodegenerative disorders, cancer cell metabolism, and cellular aging processes. The future of glycogen research spans multiple disciplines from molecular biology to clinical medicine, offering promising avenues for both understanding and treating human disease. These emerging research directions may fundamentally transform our understanding of glycogen's role in human health and disease.